manner. So let us start with today's topic. So in today's video, we will be discussing about the rhesus factor, or rather we can say that these are also called rhesus antigens. Now, why does this name rhesus come from? Now, in the year 1940, Carl Landsteiner and Wiener, they took human RBC and they found a protein in human RBCs which is also found in the rhesus monkeys. So these group of monkeys also had the same antigen which was found in human RBC. So for that it was named on those rhesus monkeys that it is rhesus antigen or rhesus factor. Now the persons who are having this antigen are called rhesus positive, and the persons who are not having this antigens are called rhesus negative. So quite both the Rh positive and negative individuals are quite normal. They don't have any problem. Now if you are having rhesus positive factor, that is when we are telling that a person is A positive. So that positive is the rhesus factor. Or when we are telling the person is A negative. So that negative factor means that the person is not having this rhesus antigen, or rather rhesus factor. So both the persons, they live a normal life, they don't have any problem. But the problem arises when a rhesus negative person comes in contact with either a rhesus positive person through blood transfusion, and in cases of female, what happens that when due to pregnancy the child of a rhesus negative mother becomes rhesus positive. So what are the problems coming when there is a positive negative clashing of this two blood groups? That only we will study today. Because if a positive person, in a positive person, a negative blood group is transfused, so what is going to happen, and how the antigen and antibody is working, so that all we will study today. So let us move on to the next part. Now in this slide we can see this is actually a pictorial representation of the Rh factor along with the A and B antigens. Now let us see what in this AB positive blood group we are already having this. I have shown you here this yellow one is the A antigen, and this blue one is the B antigen, whereas the small black square ones are the Rh factor. Now AB positive is having A antigen, B antigen as well as the Rh factor. A positive group is having the A antigen as well as the Rh factor. Now whereas B positive, they are having the B antigen as well as the Rh factor. So that is the positive part, right? And O positive is not having any other antigen, so it is only having the Rh factor on its surface. So this is a protein which is present on the surface of the RBC. Whereas when we come down to the negative blood group, that is Rh negative, these groups, they are only having the A and B antigens, but there is no Rh factor present on the surface. So in AB group, we can see only the A and B antigens. In A group, we can see only the A antigens. In B negative group, we can see only the B antigens, but no Rh factor, as it is A negative, or the Rh factor here is negative. Whereas in O negative also there are no antigens. O positive, there are no antigens, but only Rh factor was present on the surface. Whereas in O negative, neither of the antigens are present on the surface. So is it clear to everyone? So dear students, if you are not getting any part of this Rh factor clear, please do write in the comment box, ask your queries so that I can rectify or write your answers whenever I am posting my next video, so that you will be clear. So if your basics are not clear, when you are moving on with the lessons in higher classes also, in your 11th and 12th standard also, it will be difficult for you. So please make your basics clear and thorough, so that no need to learn or memorize. You just understand the things, so that when you are moving on with your lessons, it will be very easy. Okay, is it clear? Shall we move on to the next slide? So let us start with the next one. Now let us see in this slide, we are going to discuss about the incompatibility during blood transfusion. So what is happening? Now suppose a Rh negative blood group is infused or transfused to a person having Rh positive blood group. So it is safely done. Now Rh positive blood is transfused to a Rh negative person. The recipient forms antibodies in his or her blood. Now as we know that there is a body mechanism that when some antigen is transfused into our body, it will actually produce some antibodies in our blood. But when the transfusion, first time transfusion, there will be no such complications after first transfusion. But when that blood group is again transfused for the second time, that is in case of second transfusion of Rh positive blood to an Rh negative person, what will happen? Now the body has already started producing anti-Rh factors. So they start attacking and destroying the donor's red blood corpuscles. Now as they are destroying the donor's red blood corpuscle, what will happen? This blood transfusion will not only be unsuccessful, rather it can be life-threatening, because there will be a chances of agglutination or clumping in the or clotting in the blood vessels. So it is always advised that the patient's Rh factor is to be determined before transfusion. That is, if you are transfusing blood to a AB positive person, it should always be AB positive. It should not be AB negative. So here we can see in this diagram that Rh positive blood can be transfused to a Rh positive person, whether it is a male or a female person, but it should not be given to a Rh negative person, which may in turn result in some fatal conditions. So the same thing happens in case of Rh negative person. Donating blood to a Rh positive person may be fatal. But it is actually, we should be determining the Rh factor first before transfusing the blood. So let us move on to the next one, where we can see that how this is happening when a person is pregnant and there is a case incompatibility of Rh positive and Rh negative factor. Now let us see what we know about this incompatibility during pregnancy. Now suppose a Rh negative mother is carrying a Rh positive fetus. So what is happening? The Rh positive fetus, they will start stimulating the formation of anti-Rh factors or antibodies in the mother's blood, because this Rh negative mother is opposite to that of the fetus blood. So they will produce antibody in the mother's blood. Now during first pregnancy, what will happen? The antibodies are not enough. They are not so much produced that they can harm the fetus, because through the placenta, we all have already studied the reproductive system. So you all know the mechanism that through the placenta only little amount of antibodies pass to the fetus to destroy it. But what will happen in the second pregnancy? If that Rh negative mother is again carrying a Rh positive fetus for the second time, what will happen? There are already some antibodies in the mother's blood, or rather we can say that they are anti-Rh factors which are present in the mother's blood. So these anti-Rh factors which are already present during the first pregnancy, they combine with the antibodies now formed during the second pregnancy, and what will happen? The cumulative effect of the antibodies produced in the second time, they are large in numbers. So they already present in the mother's blood, and this time, second time, the antibodies which are present, they are trying to destroy the RBC of the fetus. So this causes the death of the fetus, or we can rather say that it is a state where the fetus dies, and this is called erythroblastosis fetalis. So this is a very important term you will be getting in your ICSE, that what is erythroblastosis fetalis. So I'll be discussing it with you in the next slide. So you should have a clear idea of this disease, what is erythroblastosis fetalis. Now in the next slide we will have a pictorial representation of this Rh positive mother and Rh negative Rh negative mother and Rh positive fetus, so that you'll have a clear idea. So let us move on to the next slide. Now here comes the pictorial representation. So the father is Rh positive, mother is Rh negative, and the fetus is Rh positive. So what will happen? Some few anti-Rh factors or antibodies will be present in the mother's blood, but they are not going to harm the fetus, because through the placenta only few are passing from the maternal to the fetal blood, and a small amount of fetal blood enters into the mother's circulation, but they are not causing anything to harm the baby or the fetus. But over the next second pregnancy, when the lady becomes pregnant for the second child, and here also the child is positive and the mother is negative. So what will happen? These antibodies which were already produced in the mother's body, they will attack the fetus's red blood cells, and then only the deadly condition or the erythroblastosis fetalis will happen. So is it clear to everyone? If it is not clear to you, please go through this video again and again. Try to read your textbook, whichever textbook you are following, go through it, so that make it clear, so that you can understand that how the anti-Rh antibodies are working, not during the first pregnancy, or rather in the previous slide I have told, not during the first transfusion. It is harmful when you are transfusing the blood second time, or when you are when the lady is pregnant second time with the Rh positive fetus. So this is attacking the red blood cells of the fetus, and the fetus is either it is dying through a condition called erythroblastosis fetalis, or it is rather causing some harmful effect on the fetus's body. So now let us see what is this erythroblastosis fetalis. Erythroblastosis fetalis. So this is the term we have already discussed in the previous slide, when we discussed about the incompatibility during pregnancy. So what is happening in this erythroblastosis fetalis? Now we have told already that during the first pregnancy, enough antibodies are not produced which can harm the fetus. But what is happening during the second pregnancy, when the fetus is Rh positive, the mother's body is already having some anti-Rh antibodies, and the baby again triggers the mother's blood to produce more antibodies. So as more number of antibodies are produced in the mother's blood, and they are passing through the placenta, and they are in large number, they are trying to destroy the baby's red blood cells or the fetus's red blood cell. Now this causes death of the fetus. This condition is very harmful for the baby, and in some cases it is fatal to the mother also, because she may lead to various complications. So this condition is called erythroblastosis fetalis. It is a fetal condition, and the fetus's life is at risk. So it is at times for a person who is negative to have a baby with Rh positive fetus is really dangerous, not in the first pregnancy but in the second one. So I think it is clear to all of you till now what I have discussed in this video. If it is not clear, please do ask me in my comment box, or rather if you are having any queries, write there so that I can clear it up in the next video. And dear students, don't forget to subscribe to my channel so that you can keep on getting the updates of my videos, whatever I am posting. So stay updated, subscribing to my channel, and don't forget to like and share. Thank you.